All right. Hope everyone's doing well today. Beautiful day outside. All right. We were talking about how the eye works. I don't know. We're going to talk about a lot of cool things this year, but the way the eye works is so magical. I don't know if it's appropriate for a PhD in biophysics to talk about magic, but I really find it to be one of the most phenomenal things that I get to talk about. And I just give you like the tiniest taste of it here. So last time I left us with a description of the dark current. So kind of enigmatic. And what is the dark current? So when a photoreceptor is not being stimulated, let's look at its resting state. All right, we'll call that the resting state, although it's a little bit of perhaps a misnomer. So typically, the way these photoreceptors work is in this inner segment here. There are sodium pumps, ATPases. They're ATP-driven pumps, ion pumps, that are pumping sodium out of the cell. All right, and then on that outer segment, what did I say here? Inner and outer segment. There is these sodium channels, these gated sodium channels, and they're gated by cyclic GMP. All right, when cyclic GMP is present, they will bind to these leak channels, these sodium channels, and they will be open, and sodium will flood in. So there's just a current that begins to get set up. This creates a graded potential and an electrical field in here. So there's sodium rushing in, there's sodium hurriedly being pumped out. All right, this is the dark current, called a dark current because it's in the dark. There's no photon hitting that neuron currently. And since there is this dark current that's been set up, it triggers an action potential where there is a release of neurotransmitter at the synaptic terminal of that sensory neuron. All right. Now you may be thinking, my God, what's going on here? Turns out that the specific neurotransmitter that's being released is an inhibitory neurotransmitter. I didn't spend the time last lecture describing the types of neurotransmitters. There's several categories of neurotransmitter, but there's two broad classifications. There's inhibitory and excitatory, right? So there are neurotransmitters that are going to excite or elicit a graded potential on the downstream or postsynaptic neuron. That's an excitatory one. So, for example, acetylcholine was the one we talked about in muscles, right? Cholinergic receptors get triggered by acetylcholine. But there are also inhibitory ones. And gamma aminobutyric acid, or GABA, is an inhibitory neurotransmitter. Who's heard of GABA before? Any of you? All of you? Great. Okay. Well, anyways, these are GABAergic receptors. So it's an inhibitory synapse. And it's turning down the bipolar cell. So even though there is a degranulation at the presynaptic terminal, there is it's an inhibition of the bipolar cell, and there's not a downstream cascade. All right. The other characters that we need to talk about here are rhodopsin. So that's the opsin plus the retinal molecule. And retinal, the little red thing in the cartoon here, is the chromophore. It's a conjugated pi system. We'll see what that means. That is able to absorb light. And then there's these other players here, transducin and phosphodiesterase, that we'll see what they do in this slide here. So the first step is photoreception. And the photon comes in and gets absorbed by retinal. Specifically, which retinal? 11 cis retinal. I said that it was a conjugated pi system. Does anybody not fully understand what I mean when I say that? Conjugated pi system? Okay. So when we look at this here, there's this ring structure, but then there are these bonds between all the carbons. And if you notice, they are alternating double bond, single bond. That's what conjugated means. And the electrons that are being used are pi electrons to form those double bonds. So it's a conjugated pi system. Those have unique properties, specifically because of the energy level of the orbitals, the molecular orbitals. They are able to absorb certain frequencies of energy, electromagnetic energy, specifically visible light. So they're able to absorb visible light. And this double bond right here at the 11th carbon, at C11, this is a cis double bond. Cis meaning, so double bonds, most of you hopefully will remember from organic chemistry. You guys take organic chemistry? Yeah, they are non-rotatable. You can't rotate around a double bond the way you can a single bond. And these non-rotatable double bonds have substituents, the larger substituents on the same side of the double bond. Okay. This is a little bit crowded, the stereochemically occluded, so to speak. It simply means like there's this methyl group here, and it's like poking into the chain over here. It sort of wants to be away from itself, wants to be able to open itself up. So this is a high energy state right here. It absorbs the electricity, or the light from the photon, and the light is of the specific frequency, the specific wavelength that is able to cause an isomerization. The empirical formula is the same. In fact, the connectivity and everything is the same. It's just that double bond absorbs that energy, goes to a high energy state, and then is able to relax down into the trans form. All right, so it acts as a trigger. This big tail whips out here into alignment with the rest of the conjugated system. Is everybody following that idea? Pretty straightforward. Did anyone not get that? So this is the trigger. 11 cis retinal absorbs a photon, goes to 11 trans retinal. That sets off a sequence of events. So without getting into a high-resolution analysis of the structure of opsin and the conformational changes that happen specifically in the protein, I take it for granted that the isomerization of retinal causes a slight shift, or perhaps activation is a good way to think of it, in opsin that activates an associated membrane-bound protein called transducin. Transducin, as the name would suggest, transduces this activation from rhodopsin to another enzyme called phosphodiesterase. Phosphodiesterase is an enzyme that cleaves a phosphodiester bond. It's a phosphodiesterase. It's cleaving a phosphodiester bond. Specifically, we're going to see that it's the phosphodiester bond that makes cyclic GMP go to GMP. It uncyclizes cyclic GMP to just GMP, the nucleotide. Well, GMP is unable to bind to this sodium channel, and cyclic GMP rapidly diffuses off of the sodium channel, and the door swings shut. Sodium is now no longer able to pass into the cell. For a moment, I want you to marvel at this, because our sensation of light hitting the eye and registering as something we see is so instantaneous. It seems so immediate that it feels like there could be no lag time between the moment that the photon hits the eye and we register it back in the cortex. But we're not even out, we haven't even gotten out of the photoreceptor yet. We have this isomerization happens, then an activation of transducin and an activation of phosphodiesterase, which has to then chemically cleave cyclic GMP to GMP, and then we have to, by the basic laws of Brownian motion and diffusion, diffuse cGMP off of the sodium channel, let it close enough to change the interior potential of the cell. It's a lot of molecular events that are happening right here, and we haven't even gotten to the inner segment of the photoreceptor yet. It's pretty amazing the time scale that all these molecular changes are happening on here. It's fast, faster than I can snap. So once this happens, the sodium is continued to pump out by the sodium ATPase. That sodium ATPase is hydrolyzing ATP and going through the whole process. There's a molecular sequence of events that's happening there that's shoving sodium out of the cell, but it's now no longer able to go into the cell. The cell stops releasing GABA and releases the inhibition on this bipolar cell. Now, without going into the details, which is you can get maybe when you go for a residency in neuro-ophthalmology, maybe. I don't know where they talk about it. But the synapse of the photoreceptor, the synapse with the bipolar cell and a cone or a rod, is one of probably the most complicated synapse in the body. It's incredibly sophisticated, and this is just a diagram of what's like a schematized diagram of the most simplistic manifestation that you could find it in this so-called ribbon synapse. Multiple cells are involved here. You'll remember that we had horizontal cells and amacrine cells at the boundaries of the photoreceptor and the bipolar cell, and then the bipolar cell and the ganglion cell. They are involved in these synapses because we need some sort of neuron that's going to be constitutively activating the bipolar cell. So there are these GABAergic receptors. Their glutamate is the excitatory neurotransmitter that's there. There are crosswise GABAergic receptors. There's a lot going on in these synapses that we're not going to get into in depth. But suffice it to say that the photoreceptor absorbs light, and in so doing stops releasing GABA and then releases the bipolar cell to then send a signal down the chain, which is going to pass through to the ganglion cell, out the optic nerve, and through the rest of the optic radiations. Okay. So are there any questions with that part? So that's stimulation. We have to recover. We have to recycle that retinal. We're not sitting there constantly eating carrots, and as soon as we look at something or a light goes gray until we eat another carrot, we're turning over the retinal. So how does that work? Well, you can see here that there's photoreception that happens. Retinal absorbs light, goes to 11 transretinal. The next thing that happens is called bleaching, photobleaching, where the chromophore, and this is why they call it bleaching, because the chromophore is the thing that absorbs light and gives something the observable color, actually dissociates away from opsin. It detaches itself from opsin and then goes through an enzymatic process that requires the energy of ATP. The energy from the hydrolysis of ATP to ADP is then stored chemically back, or perhaps structurally is the better word, or stereochemically, in the higher energy 11-cis retinal. It gets recycled to the 11-cis form. Once that happens, it is able to reassociate with opsin. So transretinal, what's really happening here is 11-cis retinal absorbs the photon, isomerizes, and in its isomerized form, it's no longer able to associate with opsin. It's no longer able to associate with opsin, so it dissociates off of it. Thus, opsin is free to go through the activation of transducin and phosphodiesterase, et cetera, et cetera. But then 11-cis retinal gets reconstituted, and in so doing is then able to reassociate with opsin and reform rhodopsin. So this is bleaching. When you stare at a bright light for a long period of time, or not even that long, a long period of time, and then close your eyes, you see the ghost of it, right? You see the ghost of that bright light. That is photobleaching. That is photobleaching. All of the photoreceptors, at that thing that you stared at, the bright light source, all of those photoreceptors have been bleached out, and it takes a little bit of time for them to reconstitute. Until the rhodopsin has been reconstituted, those photoreceptors are not functional, right? They're not able to establish the dark current because rhodopsin has not been reconstituted. Opsin is still free and activating transducin and et cetera, et cetera, et cetera. So until the mechanism for reconstituting retinal catches up, you're going to see the ghost of that bright light. Right? So really, the time course of how rapidly the ghost that you see goes away is due to the kinetics of the enzyme for recycling. Questions about that? All right. So, scotopic vision. The word scotopic simply means darkness. Like a scotoma is a dark blob. It's a blind spot on your eyes. I talked about this small scotoma that we all have, 23 degrees off the visual axis, laterally over the optic disc. But you can have larger scotomas. Anyways, scotopic, the word scotopic just means darkness. So, the rods are responsible for low light vision, scotopic vision. There is photopic vision from the cones. We'll talk about that in a moment. But a person who has night blindness is typically a result of vitamin A deficiency. So vitamin A is the source of retinol that is formed into retinal. So if we look at this, here is vitamin A, retinol. It's called OL, indicates that this is an alcohol. So there's an alcohol group on the end of retinol. Whereas retinal, this al over here, indicates that this is an aldehyde. It just is oxidized to it goes to a higher oxidation state, and the alcohol is converted to an aldehyde. So transretinal, then it can go through the process of isomerization. Retinol, vitamin A, the biggest source in our diet is actually from beta-carotene. Beta-carotene packs a whopping punch of vitamin A because for every molecule of beta-carotene, we get 2 molecules of retinol. We essentially just take this double bond right here in the middle of beta-carotene and hydrolyze it. We just add water across it. We add water across that double bond, and in so doing, create 2 molecules of retinol. So eat your carrots, everybody. Beta-carotene is a chromophore. It looks orange. It's what gives food their orange color, or it's also in spinach and things like that. All right. So here's the dual visual system that I was just alluding to. The scotopic versus the photopic system. The scotopic system is that which is delivered by the rods, and this is super high sensitivity. So a single photon can set these photoreceptors off, and they are monitored. Many of these rods are monitored by a single ganglion cell. So there's a lot of vertical convergence here if you think about the network that we're looking at. It's also analogous to the receptive field. Remember I was talking about receptive field on the skin with Wilder-Penfield, the like fingers versus the back. The scotopic system would be like the back, perhaps. But that's because it's monitoring about a square millimeter of the retina. That seems like that's a pretty tiny spot. Actually, it's huge. It's a large, in terms of spatial resolution on your retina, a millimeter is a very large portion of it. And that is why at night, when it's low light conditions, and the rods are the ones doing most of the photo sensing, that your resolution of fine detail is greatly diminished. Your ability to see fine detail at night or in low light conditions. At night, everything's dark; you can't see anything. But in low light conditions, your resolution goes way down because of this large receptive field. But it doesn't take much light to trigger the sensation, right? The the ganglion cells that monitor this are called M cells, big. They're very large cells, and they're responsible for returning information on motion or shadows in dim light, the general form of an object. On the other hand, there's the photopic system. Now, the photopic system is low sensitivity, but high resolution. And this should make sense to you because the receptive field is much smaller in the photopic system. However, you're going to be in more intense light to set off for the probability of a photon to land on one of these cones. So the kinds of ganglion cells that we're talking about here are called P cells. P for parvo simply means little, small. And one of the receptive field in the photopic system is only about a couple square micrometers, much, much smaller, orders of magnitude smaller. So this is giving you color, edges, fine detail, etc. There's a lot more to this. We could go into sometimes I go into the vertical or horizontal information processing, but I'm not going to do it today. I guess a little bit. I have a couple slides beyond this one, though. So because vision is such a sophisticated neural event, neural process, that the information processing that is required to be able to sensate a visual field and process it and then internally represent it in the visual cortex, it's pretty phenomenal that it's able to happen. There is a huge amount of processing that happens to that signal at every step along the way. There's a lot of visual information processing that happens before the signal ever even leaves the retina. There's a whole lot of visual processing that happens right in the retina. So, for example, there's this notion of retinal convergence. There's 130 million. So you can think of the information processing as being in two directions. There is vertical convergence, right? There's this vertical convergence along the chain here. And then there's also a horizontal integration in the other direction. We'll talk about that in a moment. But in terms of vertical information processing or convergence, 130 million photoreceptors is the typical number that is monitored by only 1 million ganglion cells. All right, 6 million bipolar cells, and then 1 million ganglion cells. So you can see that there is 2 orders of magnitude convergence between photoreceptors and bipolar cells. That's probably the biggest leap in information processing happens. But there's approximately an order or a half order of magnitude between the bipolar and ganglion cells. There's a lot there that could be unpacked, but we'll move on. Horizontal integration is where a horizontal cell at the photoreceptor bipolar junction or an amacrine cell at the bipolar ganglion cell junction is able to take information that is being encoded in the synaptic patterns at that junction and relay that horizontally to other places adjacent to it, to other synapses adjacent to it. So, for example, this horizontal cell is monitoring what's going on here, and whatever events are happening at this one, it's relaying that information to adjacent cells in one way or another, and somehow either attenuating or potentiating these synapses, which are themselves highly connected. So there's like a really high degree of interconnectivity at this horizontal, in this horizontal fashion along those synaptic junctions there. I used to do an exercise where I would go around and tell the students all that they were a color, like red, white, or blue, but it was a secret. But then they could tell, they had to guess what the image was that everybody in the class made up, but then they were able to tell the people around them what color they were, and they could hear what color. And then eventually the class was able to guess the picture that I had mapped out in the class, which was cheesily, patriotically enough, the American flag. But you can see that knowing only that, for example, you are red isn't going to necessarily tell you what the picture of the whole is. But if you know some information about the surroundings, it gives you a little bit of an insight into what the broader picture may be. Okay, so that's how horizontal integration works in roughly. Maybe it'll stimulate your curiosity at some further point in the future. All right. And then lastly is depth perception and the visual pathways. So our ability to have depth perception, my ability to see that Zoli is maybe 16 feet away from me in the back, whereas Sarah is only 8 feet from me here, is due to a couple things. First of all, it's because of binocular vision and the ability of my eyes to triangulate a distance this is a higher cortical function, by comparing the image that's coming from the left side of my eye to the one from the right side of my eye, and using the subtle differences in those images to be able to give me information about the distance of the object from my eye. So here we have, this is just showing physics of how that works. The angle in the incident angle between two objects compared from the right versus the left eye. There was we just went to go see this movie with my kids. Was it called Early Man? It's like from the Wallace and Gromit guys, and it's like claymation or whatever, and these prehistoric dudes play a game of soccer against Bronze Age people or whatever, and they're like trying to feed themselves. They get thrown out into these wastelands, and one of the cave people is trying to capture a duck. They decide that they want to like get ducks, and he like throws this rock at this duck to hit it, and the guy he's amblyopic, meaning he's like his eyes are going like this. One of his eyes is way over here. He's only kind of got one eye that he can see with properly. And so he doesn't have binocular vision. Yeah, I should see if I can get the clip for that. And he throws this rock. It's in the trailer. I shouldn't show the trailer. He throws the rock, and the rock, there's no context. You just see this duck standing there. And he throws the rock, and suddenly the rock is like traveling. It's getting smaller and smaller and smaller. And it was like a pebble bounces off this like 40-foot tall duck that turns around and bares its like gaping teeth and starts running after the caveman. He has no depth perception because he only has one eye, and he also is not able to compare. And like we who are binocular are not able to get a sense of the depth perception because there's no context to compare the duck with. It's just standing out against a sky. So anyways, visual field comes in through the back of the retina, goes through the optic nerve, and reaches what's called the optic chiasm. So one of the ways that the two sides of our brain are able to integrate this information is we take data from the left side and pass some of it to the right and vice versa. Then these these neurons synapse in the superior colliculus and in the lateral geniculate nucleus. This lateral geniculate nucleus is actually part of the thalamus. So what did I say about the thalamus? What have I said about the thalamus so far this year? What is the thalamus involved in? I mean, it does a number of things, but I've mentioned it in the Wilder Penfield lecture, right? Those slides right around there. What did I say when I was talking about the sensory pathways? You guys remember? Yeah. Yes. And what happens in the upper areas? It reaches conscious awareness. Yeah. So we don't know that something happened unless it synapses in the thalamus. For us to be aware of some kind of sensation, there needs to be a synapse in the thalamus before it projects to the cortex. And for us to be aware, it has to project to the cortex. I mean, it can do other things lower than that, but for it to reach our conscious awareness, it has to synapse in the thalamus. The lateral geniculate nucleus is the part of the thalamus that we're talking about. And you can see here that the synapse in the lateral geniculate nucleus is the last stop before it projects to the visual cortex in the occipital lobe. All right. So I'm pointing this out because we're going to see this theme again when I finally get to talk about the ear in a few minutes here. I guess I'll probably start. So anyways, are there questions on this? Yes?
So do you prey animals?
Not much. No, not much. Yeah. So but their strategy. So you know what's the? There's a little saying that eyes on the side likes to hide, eyes in the front likes to hunt. So you know predators have like a hawk or a wolf or any kind of a human. We have extreme binocular vision, like very good depth perception because it's an evolutionary advantage to know exactly how far away our prey is. Whereas an animal that likes to hide, an antelope or a horse or whatever, their survival is not as dependent upon depth perception as in the widest possible visual field. They want to be able, so our visual field is, about 170 degrees, but a horse is like, 250 degrees or something like that, or perhaps more. Bordering, in some animals, bordering on almost 300 plus degrees of visual field, having the eyes on either side like that. And that's so that it doesn't matter to them that they know exactly how far a predator is. It matters to them that there is a predator and exactly what direction that predator is in. So there's a predator over here, I can see it, and I know I need to go that way. That's what they have. They don't have binocular vision in the same way we do, for example. Great question. Any others? Okay, so I went a little bit more slowly through the eye than I intended. The eye is amazing, so I think it was worth it. The way I've organized this slide and all these lectures here, I give you the anatomy, and then I move on to the physiology, and that's what I'll do here with the ear. The primary physiology that I want to talk about in the ear is related to hearing that comes from the cochlea and our sense of motion and balance and orientation that comes from the vestibule. So I want to talk, the physiology I want to talk about is in the inner ear. So I'm not going to go, there's a lot of interesting stories to tell about the external and middle ear that I've cut out. Oh, another comment was somebody wanted to know why my slides that I present don't match the ones in Moodle especially well, and that's because why there are more in the Moodle than, and that's because I hide slides in my PowerPoint thing. I can try to be better about updating those the morning of, so that you don't have extraneous slides in there, but I also don't want you to not have slides that I may choose to talk about sometimes, which is why I've done that in the past. But all right, I did update this one though, so only the slides that I'm talking about are on Moodle right now for this. So what do we need to know here to be able to? I'm only going to go over the anatomy. You actually need to know to be able to have the discussion that we'll have obviously next time about the ear. Well, there's external acoustic meatus with your ear hole. The external acoustic meatus, this is where the pressure waves of sound come in and hits the tympanic membrane, which is like the membrane of a drum, vibrates. This vibration is transferred from across the middle ear from the tympanic membrane to the bony labyrinth of the inner ear by the auditory ossicles. The three little bones of the middle ear. I only have a minute or two here. I might as well give you guys a pearl. This is for any of you who are, because I remember somebody had said that they were really fascinated by the ear. I don't remember who that was in their comments, but I'll give you this. So in anatomy labs, when you go into a cadaver lab for, I don't know, maybe half the class here will probably end up there someday. And this will be something hopefully you remember that day. Press whoever's running that. Most anatomy labs, cadaver labs, don't actually do a dissection of the middle and inner ear. It's a mistake because it's really easy to do. And I'll tell you how to do it now because it helps tell my story a little bit. The temporal bone is the larger bone that all of this exists in. Temporal because temple, your temples, temporal means time, and your temples here is where you show the passage of time first. This is where your hair turns gray first, so they named the bone the temporal bone. The specific part of the temporal bone that the ear lives in is called the petrous temporal bone. Petrous, like Peter. You are the rock upon which I shall build my church. Peter means rock. It means stone, like saltpeter or rock salt. This part of the temporal bone, the petrous temporal bone, it's actually the densest, hardest bone in the body, which is one of the reasons maybe they don't dissect it in cadaver lab. The way to get in there is after you do the calvarectomy and you remove the brain from the brain case in the cadaver, you'll see this petrous ridge. Looks like kind of like the shape of an almond. And if you take a bone saw and you score on either side of the petrous ridge, you take some rongeurs, which are like basically bony wire cutters. You stick them in there, you pop. The whole lid will just pop right off. And put the tines in the ridges that you've scored, and you kind of gently, just enough to crack the top off. You'll reveal the entire. It'll actually look much like you see in this picture here. That the scale is a little bit off in that drawing, but you'll see the whole inner ear and the teeny tiny auditory ossicles. It's worth spending the 15 minutes, the 10 minutes to actually do that dissection if you ever find yourself there. It's pretty amazing to see the three teeny tiniest, most delicate bones in the human body embedded in the hardest little bony case of the petrous temporal bone. It's a fun thing to do. Okay, that's class.